we have recently completed a study of almost 2,500 sarcoma patients using the most powerful genetic technology, whole genome sequencing, and identified two novel pathways which are associated with an increased risk for developing sarcomas. We've developed lots of methodologies along the way, which has been fascinating. I'm happy to talk about those if you would like. But the central findings are that we've identified a pathway involved in the maintenance of telomeres, which are the structures at the end of all of our chromosomes that stop them unraveling and which shorten as we age. And it appears that patients who develop sarcomas have an inborn defect in some cases that accelerates that process. And in the second group, we've identified individuals who develop sarcoma have problems with the ways in which the chromosomes separate when cells divide. And of course, cell division is a terribly important process for all life. We start off as one fertilized egg, and we end up comprising some 70 trillion cells in the adult human. And that of course, entails many rounds of cell division. So even a small defect in the ways in which cells divide and share their chromosomes between daughter cells could result in the sort of changes that eventually lead to cancer, at least in this case. And in both of these cases, we've found that the inherited defects in these pathways was relatively unique to sarcomas, which is why we think it hasn't been observed previously. We don't see it in breast cancer or bowel cancer or other epithelial cancers. In the case of the telomere defect, we found that about 5 genes were involved, and these genes are 5 of 6 well-known members of a complex that caps telomeres called the Shelterin complex. And individuals who carry these mutations are not only prone to sarcomas, but they're also prone to melanoma and thyroid cancer, maybe other cancers. There seems to be a quite specific pattern of defects that these individuals, tumor types to which these individuals are prone. And perhaps most fascinatingly, using whole genome sequencing, in those individuals with defects in telomere function, we observed that in their peripheral blood, we could see peripheral blood cells had longer telomeres than the rest of the cohort. And this is a phenomenon that's been described relatively recently of what's called a long telomere syndrome. The defects in telomere function that we're talking about that increase the risk of sarcomas seems to be measurable in apparently normal cells in the peripheral blood, as evidenced by the long telomeres. And again, long telomeres appear to be associated with a risk for melanoma as well. So that's the essence of what I'll be talking about. And then I'll be speculating about what we can do next in terms of filling in the missing heritability in this population, because we think sarcomas are a very heritable set of cancer types. And we still know relatively little about mapping that landscape of genetic variation that leads to increased risk.